We're going to take a look at cell-mediated immunity and active and passive immunity in this lecture. Cell-mediated immunity involves cytotoxic T lymphocytes that are going to kill infected cells, cancer cells, and transplanted cells. Cell-mediated immunity is really important because if pathogens get inside of cells, the antibodies won't be able to get to them. So antibodies only can bind to pathogens that are outside the cells and the body fluids. So if the pathogen is inside the cell, cytotoxic T lymphocytes will be able to kill that infected cell. And the cytotoxic T lymphocytes are very specific for which antigen they recognize and only respond to a particular one. And the antigen has to be presented to it by another cell on what's called an MHC protein. And everyone has their own unique MHC proteins except if you have an identical twin. There are two classes of MHC proteins. We have class 1 and class 2 MHC proteins. Class 1 MHC proteins are found on just about all of the body cells. Everyone that's nucleated will have a class 1 MHC protein. An exception to this would be the red blood cells, which don't have a nucleus. They don't have a class 1 MHC protein. So the class 1 MHC protein is like an ID tag, and it's going to be presenting antigens from inside the cell to these cytotoxic T lymphocytes. And if it's one that that particular cytotoxic T lymphocyte recognizes, then it will kill that cell. The class 2 MHC proteins are only found on antigen-presenting cells, and there's only 3 types of antigen-presenting cells, so I would star this, make sure you know this. So the antigen-presenting cells are macrophages, dendritic cells, and B lymphocytes. They're going to present antigen that they've found on their class 2 MHC proteins to helper T cells to see which helper T cell recognizes that antigen. Here's a picture of the class 1 and class 2 MHC proteins. So over on the left, we've got a cell that's infected with a virus, and as the cell makes these class 1 MHC proteins inside the cell, it just randomly picks up a piece of something inside the cell and then puts it out on the surface, which the cytotoxic T lymphocytes will come around and check to see what's on the class 1 MHC ID tag. And so if they recognize that antigen, then they will eventually kill that cell. The class 2 MHC proteins are only found on antigen-presenting cells, so only the macrophages, dendritic cells, and B lymphocytes will have class 2 MHC proteins, and they will use that to present the antigen that they've found to the helper T cells. So helper T cells will check the antigens on the class 2 MHC proteins to see if they recognize it. So the antigen-presenting cells, they're going to be presenting the antigen on their class 2 MHC protein to the helper T cells. And if that particular helper T cells recognizes that specific antigen, it will start dividing and form more helper T cells and also some memory helper T cells that'll be there for the next time. The helper T cells are like the generals of the immune system. They're going to tell everybody what to do. So the helper T cells, their big job is to secrete cytokines, which are chemical messengers, and that'll give the green light to activate the cytotoxic T lymphocytes that recognize that antigen, and also give the green light to the B lymphocytes that recognize that particular antigen to go ahead and start dividing. And the helper T cells also help activate the non-specific defenses as well. So this picture just shows how central the helper T cells are to immune system function. So helper T cells are right in the middle here. You need them to activate the cytotoxic T lymphocytes or cell-mediated immunity. And you also need them to activate the B lymphocytes with the humoral immunity. And then the cytokines that are secreted by the helper T cells also help the non-specific defenses work better as well. If you take out the helper T cells and kill them off, that will take down the entire immune system, and then the person will get opportunistic infections and also increases in cancers. So the disease that takes out the helper T cells is HIV, and that's why we end up seeing AIDS in those patients if they're not treated, and they get all kinds of infections that people with healthy immune systems don't get, and they end up with lots of cancers. So cytotoxic T lymphocytes, when they find their match, they are not going to react right away. They need that double check signal from the helper T cell to say, yep, you really did find your match. So let's take a look at the cytotoxic T cell activation. So we're going to start over here on the left. We've got the helper T cell, and that has been presented antigen by an antigen presenting cell, which this could be either a macrophage, dendritic cell, or B lymphocyte. And this antigen presenting cell is going to present the antigen that it's found, maybe it's the measles virus again, on the class 2 MHC protein. So antigen presenting cells are the only cells that have class 2 MHC proteins, and they will present the antigen, in this case the measles virus, to a lot of different helper T cells to see who recognizes the measles virus. And let's say this helper T cell recognizes the measles virus. That'll stimulate it to start dividing. It's going to produce some effector helper T cells, which are going to go and direct the battle. And then the memory helper T cells will be there for the next time, and they're going to react much stronger and quicker the second time you're exposed to measles, and then you don't get sick again. And here we have a cytotoxic T lymphocyte that has been going around the body and checking the body cells and looking at their class 1 MHC protein. And they want to read what's on that ID tag, what antigen is inside that cell. And let's say that this cytotoxic T lymphocyte recognizes the measles virus. So the cytotoxic T lymphocyte will not do anything yet. It has to wait for the double check signal from the helper T cell. So when the helper T cell recognizes the measles virus, it will send out these cytokines, and that gives the green light to the cytotoxic T lymphocytes to go ahead and start dividing and kill those infected cells. So the cytotoxic T lymphocyte will divide and form more effector cytotoxic T lymphocytes, which will go and do battle and destroy those infected cells. And then there's also memory cytotoxic T lymphocytes that are produced for the next time. And they're going to react much stronger and faster if you're exposed to the measles the second time and you don't get sick. The cytotoxic T lymphocytes are going to kill the infected body cells by secreting perforins, and these proteins will poke a hole in the body cell membrane. And then the cytotoxic T lymphocyte will pour granzymes into the cell, and that'll tell the cell to do apoptosis and commit suicide. And now that infected cell is gone. And this just shows the central role of the helper T cell. And you can see that it's necessary to activate both the cytotoxic T lymphocytes and also the B lymphocytes. So it's very central in activating humoral immunity and also cell-mediated immunity. And then this is just showing a cytotoxic T lymphocyte contacting a cancer cell, and it will kill that cancer cell. The other type of T lymphocyte that's involved is the regulatory T cell. These are there to help calm the immune system down once we're winning the battle. So the regulatory T cells will inhibit the B lymphocytes and other T lymphocytes and tell them to calm down so they don't keep going and go overboard and actually cause more damage and potentially kill you. So regulatory T cells are pretty important. And the regulatory T cells also help prevent autoimmune diseases from occurring. There are 3 different types of T lymphocytes, so make sure you keep these straight. So remember, the cytotoxic T lymphocytes are going to kill any infected cells, cancer cells, or transplanted cells, and they're responsible for cell-mediated immunity. The helper T cells are like the generals of the immune system. Their job is to secrete cytokines, chemical messengers, and tell everybody what to do. So they're going to be needed to activate the cytotoxic T lymphocytes, the B lymphocytes, and then also some of our nonspecific defenses. And then the regulatory T cells are there to help calm the immune response down and suppress the activity of the B and T lymphocytes. So let's take a look at some examples of active immunity. So this is where your immune system is stimulated to respond. Your B and T lymphocytes will divide and form the effector cells and the memory cells. So an example of natural active immunity is when you get an infection. So maybe you get sick with the flu. And memory cells would be produced because your B and T lymphocytes were stimulated to respond. Artificial active immunity, anytime you see artificial, you know that a needle is involved. And a way to artificially stimulate your immune system to respond would be to give a vaccine. So maybe you have the MMR vaccine. And so you will produce memory cells that will protect you if you are exposed in the future to measles, mumps, or rubella. And this is what happens when you're given a vaccine. You are either given a killed pathogen, or you could be given one that's alive but weakened, is not going to cause the disease, or you may just be given parts of the pathogen. So that vaccine will stimulate the primary immune response, which remember that takes about 10 days, and you will stimulate the B and T lymphocytes to start dividing and form the effector cells. So for example, the plasma cells from the B lymphocytes would be produced, and you would be producing antibodies, but you don't need those right now because you're not really infected. But what's important that's produced are the memory cells, and so they will be long-lived, perhaps the rest of your life. Some of them are shorter, and they will protect you in case you are exposed to the disease later on. And then if you are exposed later on to, say, the measles, then those memory cells are going to respond very quickly and very strongly, and the secondary immune response, you don't get sick. Passive immunity is when you get antibodies made by someone else. So your immune system is not activated and you don't produce any memory cells. A way to naturally get antibodies from someone else with natural passive immunity, there's two examples. One would be passing IgG antibodies made by mom to the baby across the placenta, and so that'll protect the baby for the first 2 months of life. And then the other example of natural passive immunity is mom passing her IgA antibodies in the colostrum, that first breast milk, to the baby. So no memory is created because the baby's immune system was not stimulated to react, and so there won't be any memory B or T lymphocytes made. And mom's antibodies last around 2 months, and so that's why we start vaccinating the baby around 2 months of age for a lot of the vaccines. And then artificial passive immunity is going to involve a needle, and how you can artificially give someone antibodies made by someone else. There's a couple of examples. Let's take a look at the next picture. So if you've got tetanus or botulism, for example, those bacteria produce a toxin that cause the symptoms of tetanus or botulism. And so if you are suffering from those diseases, they would give you antitoxin. So they're going to give you antibodies, which are called antitoxins, that will bind to the bacterial toxin and prevent it from doing more damage to your body. If you got bit by a rattlesnake while you're out hiking, they will give you antivenom. So you're being given antibodies that will bind to the rattlesnake venom, which will help prevent the snake venom from doing more harm to you. And then another example of artificial passive immunity would be being given an immunoglobulin shot. So if you were exposed to hepatitis B virus, maybe by a needle stick, and maybe you hadn't had the hepatitis B vaccine, they would give you the immunoglobulin against the hepatitis B virus. So they would just give you antibodies that would bind to any hepatitis B virus in your body, and then that would prevent it from getting into your cells and also mark it as a red flag for the other white blood cells to come around and get rid of that virus. And there's no memory that's produced with artificial passive immunity because your immune system was not stimulated to react, and so you didn't divide and create any memory cells. So these antitoxins, antivenom, immunoglobulin shot will last for a couple months at most, and then they will disappear, and then you'll be vulnerable again.